We often study human diversity in terms of culture, ethnicity, and psychology, rather than raw biology. The taboo around discussing biological human differences in species-level terms makes science cautious, sometimes too cautious in exploring the possibility of hidden divergence. We also measure humans against our own definitions, which are biased toward keeping us in one box.
When humans control and stabilize their external environment, evolution can shift inward towards immune function, hormone regulation, metabolism, or it can change how we process information, form memories, and perceive time. Evolution would also alter gut bacteria and skin flora, and it would alter cell efficiencies such as their energy usage, repair rates, and sensory filters. If human evolution has evolved from how we look on the outside to how we develop our insides, how would we notice?
We could be missing the moment in history when humans begin truly diverging, simply because it's happening in the invisible parts. Once a species can build shelter, clothing, tools, and tech, it no longer needs to grow fur, bigger claws, or heat-resistant skin. So evolution's pressure shifts inward. Brain, perception, pattern recognition, energy efficiency, and cooperation strategies. So, is adapting on the inside phase 2 of evolution?
There are certainly cracks forming in our current understanding. Medicine, technology, and global travel hide differences that would otherwise lead to natural reproductive barriers. Microbiome composition, hormone regulation, and brain wiring can diverge without showing obvious external traits. Fertility between some human groups hasn't been deeply studied at a biological compatibility level. We once bred with Neanderthals and Denisovans, but they were different species. Interbreeding doesn't mean sameness. So now, we're selecting when to use different sets of rules to measure the same things. It's not going to make sense. If we strip out the human self-labels and just look at it like a microbiologist watching bacteria evolve inside a host, the picture changes dramatically. We're walking, talking ecosystems where the human cells are just part of the habitat. Some microbes directly influence mood and cognition. They can change immune calibration, which decides how reactive the body is to allergens and infections. Microbes can influence nutrient extraction, which means some people's microbes pull more calories from the same food. And then there's toxin handling. From a microbial perspective, a shift in the dominant community inside the host can subtly change the host's behavior, mood, and even social patterns.
From the microbial perspective, what matters is what food you eat, what pathogens you've evolved with, how your microbes talk to your brain and immune system, and whether your microbes could survive in another human without collapsing. If we looked at humanity purely through microbial compatibility, Homo sapiens would already be several distinct biome species, and interbreeding between them would require microbiome transfer to succeed long-term. This perspective challenges a deep but unexamined assumption in anthropology, evolutionary biology, and medicine, that humans can be understood mainly through human DNA while treating the microbiome as a side note.
Most species definitions ignore co-evolved microbiomes, yet in humans these microbes directly affect digestion, immunity, mood, and possibly consciousness. If the microbiome is part of the functional unit of a human, then divergence in microbiomes is a form of evolutionary branching. It could explain health and compatibility puzzles. Some cross-population health problems such as autoimmune disorders, pregnancy complications, diet-related illness, may have microbiome mismatch as a hidden cause. This opens a new research direction for studying biome compatibility between human groups, not just genetic compatibility.
In fields like transplantation, reproductive medicine, psychiatry, and nutrition, microbiome differences could be the missing variable that explains why some interventions work in one population but not another. We may need to preserve microbial diversity in isolated groups just as much as genetic diversity.
Should species definitions for complex hosts include their co-evolved microbiomes?